Okay.
Well, welcome to the College of Complexes. This is our 428th meeting since we started in February of 2009. We put a different speaker on every week, a different subject. Our speaker has to be for or against something. We don't care what it is. We give him an hour to make a presentation, up to an hour. If he goes over an hour, we cut him off. If anybody interrupts the speaker, we remind the interrupter. We listen only one fool at a time, one of our rules. Then we have questions and answers from the audience, not speeches of the speaker. Then we have remarks, rebuttals. Everyone in this audience that wants to gets 5 minutes at the podium here to respond to what the speaker said, for or against. And our speaker is the last word. He gets comment, close the meeting. That's how it works. But before we introduce our speaker, we have time for announcements. Anybody have any announcements they want to make? Now's the time. Any announcements? Margaret, anybody? Nobody has an announcement? I can't believe it.
There's one here. On the 25th, I believe, is the Philosophers Forum next meeting, and we have a speaker dealing with the woman question. I think it's a nurse that's making the talk. But if you're a woman and you've got questions about the woman question, you're welcome to come.
That's a big question.
Any other announcements? Now's the time. Oh, no more announcements, okay. Any announcements back there? No? All right. Well, next week, we're going to have Calvin Bluet. He's going to talk about why America needs a new foreign policy principle for the 21st century. That's on the itinerary here. I'm not going to read all this. You've got a copy. On September 29th, we're going to have a speaker talking about comfort women. They are victims of Japanese military sexual slavery. And uncorrected outrage with sex trafficking to this day, which shouts for rectification. It's 2 weeks from now on September 29th. Then on October 6th, Michelle Valentino is going to talk about on why identify with Beto O'Rourke. She is for Beto O'Rourke, I guess, so you should read that. Anyway, you may learn something. Our speaker tonight is Fred Grinnell. I believe I said that right. PhD. Robert McLennan, Professor of Medical Science, Department of Cell Biology, Ethics in Science and Medicine Program, UT Southwestern. Quite a pedigree here. We'll discuss how genetics changes the practice of medicine, the challenge of genetics to human research, ethics, especially involving embryos, and the historical context of eugenics. He will argue and conclude that in the larger sense of predictive genetic medicine, we are not our genes. Without further ado, please give a very, very warm welcome to Fred Grinnell. Yeah, I will.
Good. Okay, so the most important thing is for you to be able to see the slides. If you can't see the slides, then you're going to miss probably a lot of what I have to say. So if you can move a little, huh?
Breaks and center it.
Just turn it sideways a little bit. There you go.
How about that? Okay, so we can get rid of this. We can get rid of those. I'm used to talking to 200 medical students at a time, so I don't think I need microphones.
Right on.
And
Are they filming you though?
and they don't need to film me, they need to film the slides. Because my voice is what explains what the slides are, but the slides are the key. And actually, if you all wanted to just pull your chairs into the middle here, you could do that. If you wanted, can you hear me? You can hear me, right? Okay. All right, so I'm happy to be here today and talk to you about a subject that I teach about that interests me a lot, which is modern genetics and the ethical issues that modern genetics raises. And you already heard what the discussion topics are, how genetic medicine changes, medical talk, about disease, patient, and cure, the ways in which these changes challenge human research ethics, and then we'll sort of put these ideas in the context of what's called eugenics. And I'll have to explain to you about that. So I became interested. The trouble is the putting it on the side is a little bit out of focus. Can we focus this better, do you think? Yeah. Is there somebody here who's a good focuser? Must be. Oh, wait a second. Here we go. Maybe this focuses.
You
Is that
have,
better? No. I think this is focus. This is the focus device, right? Here, right here? Yes, this is focusing. And if we took it back further, would that... See, it has to be sort of... You know, and it's going to have to be over on the side there because otherwise, if it's on an angle...
Here's what you can do.
So the more you can move it over, but you can't aim it in the middle because then it's all out of focus. So I became interested in these issues that I'm going to talk about today. Quite a while ago, we got a grant from the Reynolds Foundation to study heart disease in relationship to genetics, and that was a very early application, and we had to come up with a consent form to tell patients what they needed to, what it was going to be like, and we had to figure out what to tell them about risks and benefits, about who's going to get to see their genetic data, about what happens if we find something about later, and are we going to tell them about it? And so that was my introduction to the whole issue, these issues. And you'll see that these questions that I mentioned here have just finally have been settled in terms of best practices at the level of federal oversight. So up until now, it just has been sort of, everybody's been trying to figure it out as it goes. So what I'm going to talk to you now is I wrote about a few years ago in a book that I published called Everyday Practice of Science. And this is a kind of update on chapter 4 of that, chapter 5 of that book. So genetic medicine gets used for potentially a lot of different things. And here are some of the clinical uses listed here. It's very important for identifying disease subtypes. So if you have some kind of, let's say, a tumor, it can be very helpful for figuring out about that. It's important for pharmacogenetics. If you have certain genetics, certain drugs will not be right for you. It's good for tissue typing for transplantation. And then it's good for a whole bunch of things in terms of identification of risk for adults, for newborns, for parents before conception, for moms after conception, and for embryos that have not yet been implanted. So if you think about this long list, you can think that there are actually two kinds of genetics going on here, genetic medicine. One is for people who are sick. They already have symptoms. And the other is for people who are healthy. They don't have any symptoms yet. And so where I'm going to focus my attention is the situation here. For the pre-symptomatic folks, the folks that don't have any symptoms, so up here we call this precision medicine, but down here this is predictive medicine. So we're really focused today in this conversation about predictive medicine. Okay, so predictive medicine changes what we mean by disease. So let's think about it. 100 years ago, none of us were here probably, but if we were, and we felt sick, what we would do is we go to our healthcare provider and say, hey, I feel diseased. Hey, I'm the one who knows that I don't feel well, and I'm going to try to do something about it. But I'm the one who knows first. And then we get modern physiology and anatomy. And now we can start thinking about disease as being atypical physiology or atypical anatomy. So now I'm feeling great, and I go to my physician, and he says, well, Fred, let me do a few tests. And he comes back a couple of hours later and says, oh, your blood pressure's too high, you know, your cholesterol is too high, your bone density is too low. I came in feeling great, and I leave feeling really sick. The point is, though, that what's happened is that I haven't felt diseased by these symptoms or these features, but they're already there, and they're atypical. But what happens in genetic risk is that I don't have the symptoms. I just have the possibility of the symptoms happening. So I can start talking about disease even before, for an embryo, even before there's a body plan. Disease now is risk about something in the future when we talk about predictive medicine. That's totally different. But it's a natural consequence of the way that we do research. So we knew that, you know, you were diseased. Well, how come? And we found out you had some abnormal physiology. Well, then we started studying the genetics, and we said, hey, there's a genetic difference that could account for this. And once you have this pathway, then you can just turn it around. And now we can test and say, hey, if you have this difference, you might develop this physiology, which means you might become diseased. So that's what's happened. And once you have genetic tests that can make predictions, there's a whole industry that's developed. And some of you, many of you have probably heard of the most famous company in the United States, which is what?
Walmart.
Huh? It's not Walmart. Famous genetic testing company. So, Ancestry is, there are two of them. Ancestry.com is one, and 23andMe is the other. Okay, so there are now hundreds, literally hundreds of companies that offer genetic testing to people who are interested. Now, why are people interested? What makes people so interested? This book is a good example of what's happened and why people are so interested. This man who wrote this book, Francis Collins, is the head of the National Institutes of Health. It's called The Language of Life. You can't read it, but up here is this little byline which says, your life depends on the secrets of your DNA. Are you ready? And what that does is it leaves people thinking that you are your DNA. That's the impression that the medical community, including that I'm part of, has given to people, you know, who are not part of medicine. And so we call this the personalized genomic medicine. It promotes a belief in the idea of genetic essentialism. Genes are what count for most. And you look at Time magazine, and you can see on the cover, you have the IQ gene, or you want to know my future, you can just look at all these different genes and you can figure out what it is. But that's really not the way it is. So epidemiology talks about what they call the 3 laws of genetics. You know, like the 3 laws of thermodynamics. Well, these are the 3 laws of genetics. The first one is that health and health-related traits are influenced by heredity. The second one is that the effects of being raised in the same family is smaller than the effects of genes. And the third one is that the effects, for most diseases, the effects of life experience after leaving home is much greater than the first 2 combined. So if you think about most diseases, not all, but most diseases, let's say it's 75% what you do after you leave home, 15% what your genes are, and 10% what your home life was like. Now that's not all, but that's most. Okay, so that's a whole different image. You are not your genes, you are your life. So if you ask the CDC, what the Center for Disease Control, what genes we really need to know about right now. Remember, the average estimate for the number of genes we all have is something like 20,000, roughly. How many of those do we need to know right now? The CDC says they call these tier 1 genes. There are 3. You can't read it. Okay? But anyway, one of them is the gene, the genetic sequence associated with BRCA1 or 2, which are hereditary breast cancer genes for women. There's another that's associated with another type of cancer, and then the third is associated with what's called hypercholesterolemia, where you have unusually high cholesterol levels. So those three genes are it. Those are the type 1. But and the reason why they're identified that way is because of their early detection and intervention could significantly reduce morbidity and mortality. So that's what makes them so important. They really are, in some key ways, related to our future health. Now, the ACMG, which is the American Society of Medical Genetics, actually recommends a whole bunch more. They recommend 59 genes, and they do so because they're all important, but they're not as important or predictive for medical well-being, but they're not as manageable as the ones that CDC refers. So the biggest genetic study that's about to occur, or just started in the United States, is called All of Us, and it's being run by the National Institutes of Health, and they want 1 million genomes. And that's what they're hoping to get. And one of the things they just decided is they're going to give you back 59 disease variants if you participate in this. And there are a lot of folks who are concerned about giving back so much information, how people are going to use it. So what are the risks? What are the risks of learning your genetic information? So,
The insurance companies.
well, we're going to talk about insurance companies in a minute. But the number one risk is you might learn something about yourself that you wish you didn't know. That's the number one risk, and that learning that thing can lead you to change your behavior unnecessarily, to spend money on other kinds of things unnecessarily. So that's number one. Number two, you can start asking questions. Is your genotype worth marrying or reproducing or leaving an inheritance? Is your fetus's genotype worth continuing the pregnancy? Is your embryo's genotype worth implanting? Your child's genotype, and this is a big deal, might show that the husband is not the father. You know, what they say is the mother is a matter of record, the father is a matter of opinion, and that turns out to be the case in a lot of cases. And one of the things genetics has shown is that probably 1 out of 15 kids, who the dad is not who they thought the dad was. Now or in the future, your DNA might show something about you that you don't want to know. Your extended group or family might learn something about its origins that you feel that you all would not like to know. Your DNA data might not be private. You may have trouble getting life insurance or some other things, and I'll mention that more in a minute. So we do have what's called the Genetic Information Non-Discrimination Act, and that protects our medical applications. So somebody cannot deny us medical treatment because they know what our genes are. Okay, so that's health insurers. But there's no protection against life insurance or disability insurance or long-term care insurance. People can use the genetic information to regulate that. Now, employers can't use our genetic information as long as they have at least 15 employees. But if they have less, they can. And of course, there are no rules about research, although there's some changes going on. And there's no privacy protection. Here's one example of one guy who wishes there were privacy protection, but the rest of us are probably happy they're not. So this guy who was a serial killer back in the 70s, they had samples of his DNA, and they didn't know who he was. But somebody said, hey, wait a second, why don't we check out his DNA against Ancestry and some of the other companies that do, testing, and they found some sequences that were similar. Not identical, but similar. And so they went to the people who had some relationship, and they asked them, well, do you have any cousins or relationships that might... And one of the people had a fourth cousin or third cousin, I forget what it was, And they gave them that information, and then the authorities were able to get a genetic sample from that guy, and they tested it against the original associated with the murders, and they caught themselves the Golden State Killer. And so that's great, but there are a lot of people where, well, you know, our information is out there. People can use it for everyone. Every person in this room, I'm guessing, every person in this room has samples in a laboratory, in a hospital somewhere. We've probably all been to hospitals. We've all had maybe a pathology test or something done where there was some tissue sample of ours. It's there. In 1999, there was a study that I'm sure it's been a more recent one, but this was convincing enough, that there are 265 million pathology specimens representing 65% of the U.S. population. That was 1999. I'm sure it's gone higher. Only now, which you can't read here, has finally the federal government changed what is called the common rule to deal to some extent with this. So if you read over here, it's just slightly out of focus. Oh well, let's try. This is all of these names here are names of different departments in the government, which is why they call it the common rule. And these are the rules that regulate all of research, and they've just changed the rule so that now it has the requirement, which you can't read, that if you're going to have to tell somebody whether or not their samples or whatever they get as part of the research will be used it will be used for research purposes, even if they take your name off of it. So you have to give them permission, or they tell you that nothing's going to be used, and you have to decide where you want in that whole arrangement, and how you want to change things, and then they have to give you other information which you can't read about whether there's going to be any genomic testing involved and things related to genetics. So the rules are now changed, and they finally answer, in terms of guidance, those are initial questions that I posed about the original study that we started. Okay, so we talked about disease. Let's talk about patient bodies. So right now, in conventional medicine, we have three major types of bodies. We have the fragmented body, because each of us has a different doctor for a different thing. We got psychiatrists, podiatrists, cardiologists, orthopedists, you name it. And nobody takes care of all of us. So we're pretty fragmented in terms of how medical care, the medical system treats us. And then we have mosaic bodies, where we have pieces of other people inside us, heart transplants, kidney transplants, liver transplants. And we have brain-dead bodies on external life support. What these bodies have in common, however, is that one patient equals one body. But genetics changes that. Now, in genetics, we have two kinds of new human bodies. We have two patients. We have the many-body patient. Because genetics doesn't just look at the individual, it looks at all of their siblings and relatives as well, like in the case of the guy who just got caught. And we have the embryo patient. So we have one embryo, one patient. One patient, many bodies is because if you know her genetic information, you essentially know everything about her twin's genetic information. So the question is, what responsibility does she have? And in medical care, privacy trumps autonomy. So if she finds out that she has something really serious genetically, she doesn't, and her physician doesn't have to tell her twin sister. So her twin sister's autonomy is not as important as her privacy. Okay? That's for medical care. It's different in research. In research, her autonomy is more important, it turns out, or we'll see if it's going to stay that way. than her privacy. Because in order to test this person's genetics, you don't have to get her sister's permission, even though testing her is going to turn out to tell you everything about her. So some of you have seen this book. It's a great book. If you haven't, The Immortal Life of Rebecca Skloot. So in this book, they talk about Henrietta Lacks, whose a sample of whose cells became known as Hela. Henrietta Lacks Hela cells, which have played a very important part in medical research and development of different kinds of drugs. And they have, and at the beginning of it, it talks about what happened and what the ethical issues were with what happened to her. And it starts out by saying she gave permission, to have to be treated at Johns Hopkins. This is back in the 1940s, I think. Nobody told her that, or asked her actually if she wanted to be part of research, but somebody took a sample, and probably that would have been typical of what goes on now. And then the person who took the sample, the pathologist, wrote in the chart, the patient tolerated the procedure well and left the operating room in good condition. And on a separate page, he wrote, Henrietta Lacks biopsy of cervical tissue given to Dr. George Gay. And the problem with this, the ethical problem with what happened, is that her name was there. If her name had not been there, which now it couldn't have been there, but if her name was not there, there would have been no book. Because even though somebody might have called them Hela cells, and even though it might have had all of the impacts on modern medicine that it's had, nobody would have known whose cells they were. And you didn't need to know whose cells they were for all of the uses to which those cells were put. Okay, so now, a few years ago, researchers wanted to publish the genome of Hela cells, and the family said, the living family, oh, wait a second, that's our genome. I've got a little privacy here. And they threatened to sue the federal government and NIH. And nobody wanted to really test this in the courts because nobody's really decided yet whether or not you have any privacy. And so there was a deal done. This is Henrietta and her husband. And basically what there is now is a committee at NIH that has oversight of access to the information about the Hela genome, and specifies who can have access to it and not, depending upon whether or not what they want to do fits in to a set of agreed upon types of research and uses. Okay, so that's the multi-person patient. How about the embryo? Well, the embryo, you probably heard, it's getting used for everything. IVF, genetic testing, stem cells, therapeutic cloning, gene therapy. The embryo is such a good genetic patient because it's all gene. It's almost no history. So when you make changes to the embryo, it results in changes to everything else that happens subsequently. If you wait until you're a lot older, then the blue is all history, and there's not all that much left that you can change that will influence everything subsequently. So the genetic options decrease as the embryo body becomes the historical person. So embryos were first isolated in 1970. Or I should say prepared, not isolated, in Britain. And several years after that, in the United States, we had the famous Roe v. Wade case that legalized abortion. And the opponents of abortion saw in embryos an analogy. And they did not want embryo research to go on because they felt like when you destroyed an embryo, it was like an abortion. It was analogous to abortion. After Louise Brown was born, the first embryo baby in 1978, there was again a discussion, and actually a committee was appointed at NIH to recommend whether or not we should be using federal funds to study IVF in embryos. And the committee said yes, but there was so much political pushback, it never happened. And in 1995, a famous amendment to the NIH, the federal budget, called the Dickey-Wicker Amendment, said that it's illegal to use federal funds in the United States to do anything that would harm embryos. And that's the way it is from then to now. So there is no federal research in the United States that goes on, federally supported research, which is the majority in the United States that supports research if embryos are going to get damaged. So what is the embryo anyway? And this is a big question, because if you think about it, embryos never existed outside moms until 1970. Up until 1970, embryos were part of normal reproduction, not in test tubes. So what is the free-living embryo? And what's the difference between the embryo body and the embryo person? And when does the moral status of personhood begin? So there are two views. One that consistently the Catholic Church expresses, which is the most consistent, I have to say. Not that I'm saying that I agree with it, but is the most consistent, is the ontological perspective. It's there all along. The embryo is the person not yet born. And the other is the existential perspective, which says that it emerges over time. So what does that mean, emerges over time? Well, part of the reason why the Catholic perspective is so consistent is because they can say, well, embryo formation, you know, is when it begins. Everybody else wants to say, well, it's day 14, when the primitive streak forms, because that's the last day that you can have twins developing. After that, you know how many people you have. Or it's day 40, so-called quickening, which was what led the famous Greek philosopher Aristotle to say that's when the embryo first really has life, and when a number of religious groups said that's when ensoulment occurs. Or down here around day 24, when you have independent viability, or over here when you have birth, separation from a mother, or some philosophers who are, you know, who are sort of out on the edge say, well, the embryo doesn't really have any moral authority until it has moral agency and make its own moral decisions, which is like when they're about 22. Okay, so we don't know what the embryo is, and that debate continues. But there are a lot of applications, and here's one of them. We now have 3-person embryos. This is one of the latest techniques. What happens is that here's a mother's egg, and this mom has unhealthy embryos, unhealthy mitochondria. That's a structure inside the egg that is involved in producing energy. And here's another mom who's going to be a donor, and she has normal mitochondria. So you take the nucleus out of the donor mom's egg, stick in the nucleus of the mom who wants to have a kid's egg, and now you have the mother's nucleus in a donor egg. This is surrogate ovum therapy, that's what I want to call it. Although that name hasn't caught on, I have to say. And now dad, you know, fertilizes his egg. So now you have parents, for the first time, we have parents, we have kids. We have the first kid born 2 years ago who has a DNA from 2 moms and his dad.
The question is for mitochondria DNA.
I'm sorry?
It's only the mitochondria DNA that is from the donor.
Yes. Yes. Yeah. And we had a major study at the National Institutes of Health that tried to evaluate, is this okay? And the answer is yes, but there's no research on embryos federally funded because FDA is federally funded. And FDA can't approve new technologies because of their restriction on federal funds. And now the most recent thing is genetic genome engineering with what's called CRISPR Cas9, where actually you can, this enzyme called CRISPR with a guiding RNA can actually go in and change the DNA itself. one base or one gene at a time. So this is fantastic. And UK approves the first studies of new gene editing technique with CRISPR on human embryos. This was a couple of years ago. We had our study in the United States. It says it's great to do as long as we have the right, set of restrictions and oversight. This time Congress passed an extra set of legislation to prevent this from happening, because Congress said this time that if somebody sends in an application to do this, you can't even acknowledge that you received the application. It's not even that you can't acknowledge and say, I'm sorry, federal funds can't be used for this. You can't even say that. All right, so who's doing it? Well, we're developing the technology, but China is doing the medical stuff. China gets another CRISPR first. This was a piece, an essay in Wired a few weeks ago. China, I can't read these, but these are a whole bunch of clinical applications that China is developing with this new CRISPR technology.
This is to get rid of some bad gene to do this?
Yeah, so, or add a good one. So a few years ago, a number of years ago, 1972, this book appeared, The Ethics of Genetic Control. This was really forward-thinking stuff. So this guy Fletcher talks about euphenic medicine, which is a word that I had never heard before and rarely since, which is just using normal medical techniques to treat genetic diseases. Eugenic, which is encouraging or preventing a genetic condition through selective breeding or termination. We'll talk about that. And genetic, changing the genes themselves through deliberate modifications. That's what he was predicting would happen, and now here it is 45 years later, and it is actually happening. And some people don't like to talk about this because it ends up being, well, we're going to repair the genome spelling mistakes, okay? And so gene therapists become editors who amend, delete, correct the defective text of disabled bodies, and that suggests that flawed genetic text is associated, that disease and disability means defective. And that's a real scary aspect of eugenics that I'm going to talk about in a couple of minutes. So euphenic interventions now, what do we have? We have euphenic interventions. You've heard of people who have PKU, and they go on a special diet. If you're unfortunate and have the BRCA1 or 2 mutations and you're a woman, you can reduce the risk dramatically of breast cancer by having your breasts removed. We have genetic interventions I've just talked about, like gene therapy or this mitochondrial gene replacement or the CRISPR technique, and eugenic interventions, which is where we want to talk about now, which is avoid the marriage, don't have children, don't implant the embryo, or abort the fetus. Those are all eugenic interactions. So it's a big business now to do pre-implantation genetic diagnosis. This company called Counsel, you see here an embryo, they're going to take one cell, they're going to check the genetics of that cell and decide whether or not to implant the embryo. And then there's this major discovery made a couple of years ago that you can find in mom's blood genetic signatures of the fetus. So what that means is that you can analyze the fetus just by taking a blood sample from mom. And there are a whole bunch of companies that do that now. So they will take a sample, a blood sample, just a blood sample, and they can tell you, for instance, whether you have trisomies, such as are associated with Down syndrome.
You want some water?
No, I've got one here.
Okay.
Thank you so much.
Somebody back there is calling for water for you.
So I have a special affinity for this one here, Natera, because my son works for them. So he goes around selling this stuff, and I go around discussing its ethics, and we have interesting conversations. The disability community doesn't like these tests at all because, in part, what happens is that people who have things like Down syndrome, where there's a positive diagnosis, end up often having the pregnancies terminated in a very large percentage of cases. And the idea develops that preventing the disability means preventing the person. And that's what they don't want to hear. What they want to hear is that disability often is a social impairment, and so we should be focused on trying to not prevent the person, but treat the disability. And it's a really important distinction that community makes. So if you think about genetic condition, biological impairment, social disability, you become blind, or you're born blind, or you lose motor, you're wheelchair bound. These are clearly biological impairments. But depending upon what society does, they can be more or less disabilities. So a blind person can often live very, very well and say actually they can experience life in ways that somebody who's sighted cannot. So it's so that distinction is very important. Now, so we have things like Down syndrome or cystic fibrosis or Huntington's disease. Those are clearly biological impairments. And right now, to a significant extent, they're social disabilities. But the question is, can we have interventions that will treat those diseases and make it possible for people to have lives that aren't socially disabled, notwithstanding the biological impairment? And then, of course, we have imposed disabilities. So gender, race, ethnicity, it's not a biological impairment, but socially we can impose disability. And when we do this kind and stop making this distinction, the biggest concern is we stop funding and supporting research to try to correct these disabilities. So for instance, this was a feature piece and just a couple of weeks ago. It was either in Science or Nature, and it was about a new treatment that's developed. Patients thrilled to reports of a promising antisense drug against Huntington's disease. No one's sure yet whether it will work. Well, the point is that if you have the Huntington's gene, you know that you're going to eventually get Huntington's disease. It's 100%, although you don't know exactly when. If you're going to just abort all the embryos or all the fetuses that have Huntington's disease, then we don't have to find research to try to fix it. But there have been a lot of fantastic people who had Huntington's disease who lived very long, productive, society-rich lives until they developed these diseases in maybe their 50s or 60s or 70s. So, and gender as a disability is not going to go away unless we change our attitudes. That's what you're talking about. What'd you say? You're talking about a philosophy form, the gender problem? Well, there is, not in the United States, obviously, but around the world, there's a global war against baby girls. You can't, right down here is where the relative number of boys to girls would be under normal circumstances, and these are numbers showing in different provinces in China, very large increases in the number of boys, and here is similar kind of results for many countries around the world. So girls are discriminated against, and when there's a pregnancy involving girls, and this has been called gendercide, which is an interesting name, and this is from the Kathmandu Post. Because Tibet legalized abortion. And as soon as they did that, the number of boys to girls births increased dramatically. So that's what this article is about. So a few years ago, the Office of Technology Assessment, when the Human Genome Project started, wrote that new technologies for identifying traits and altering genes makes it possible for eugenic goals to be achieved through self-selecting technologies as opposed to government-imposed social control. And that's potentially, what could happen. And especially if you think about the impact of books like The Language of Life, Your life depends on the secrets of your DNA. Are you ready? So this genetic essentialism feeds you genetic impulse. It's like you put each person under a microscope, you check out their genes, and if you don't like your genes, you pull the lever and goodbye. Okay, and so that's the view of eugenics. So eugenics was a word, it means good genes. And it was an idea introduced by Charles Darwin's cousin, a man named Francis Galton. And Darwin's cousin, Galton said, listen, we spend so much time trying to breed the best horses and breed the best cows. Why don't we spend more time trying to figure out how to breed the best people? Okay? And then this guy, Alfred Plutz, took this idea further, and instead of thinking about breeding the best people, he thought about getting rid of the worst people. Okay? He called that racial hygiene. Racial hygiene. And okay, if you've got some disease, and we treat you and make you better, you're just going to pass it on to your offspring, and they're going to have the disease. What we need to do is stop making people better with medicine who are diseased. Okay, and that way we're going to make the population much better. Well, in the 1920s, well, actually in the early 1900s, the U.S. philanthropic community loved this idea. All of these companies, Russell Sage, Carnegie, Rockefeller, Harriman, Kellogg, all of them were putting big money into eugenics because this seemed like the way to go. Okay, so this is the progressive application of science to eradicate social pathology. It seemed that nobody occurred to anybody that social pathology might be a result of social conditions. So we're going to attack the pathology that's source by eliminating effective genes. And that was what the goal was. And we don't want charity, because alleviating human suffering is unenlightened and inefficient. And if you think about it that way, it's not the right thing to do. So, and in medical school, there are people who wanted to teach eugenics. The place of eugenics in the medical curriculum. This is Harvey Ernst George, UVA professor of anatomy, and then later dean. The ultimate idea is the perfect society, elimination of physical, moral, and mental sickness, and doctors are the ones who are going to be in charge. So we need to be teaching eugenics. And some of his students, these three guys, Hugh Cumming, who became the U.S. Surgeon General, Talia Farrow Clark, and Raymond Bondelier, were responsible for the greatest research program that lacked any kind of ethics in the United States, called the Tuskegee Research Program. Something you may have heard of. That the center of eugenics in the United States was at Cold Spring Harbor, which is one of the major research institutions now. Here's the annual meeting of the Eugenics Research Association in 1918. And that society pushed a program that was you could define, according to this statement, the character of a nation is determined primarily by its racial qualities. That is, by the hereditary, physical, mental, and moral or temperamental traits of the people. And they were very much involved in immigration policy. This bill, which limited in the 1920s immigration, the Immigration Restriction Act of 1924, which was aimed to halt the immigration of dysgenic Italians and Eastern European Jews. And they promoted eugenical sterilization across the United States. So eugenical sterilization actually reached the Supreme Court in 1927. So in this famous case of the state of Virginia versus Carrie Buck. And they wanted to sterilize Carrie Buck, and her lawyers were trying to keep her not from being sterilized. It got all the way to the Supreme Court, and Oliver Wendell Holmes wrote the response: "It's better for all the world if instead of waiting to execute degenerate offspring for crime, or to let them starve for their imbecility, society can prevent those who are manifestly unfit from continuing their kind." That's our Supreme Court in 1927. And after that decision, sterilization became practiced in many states. There were 60,000 Americans, mostly women, sterilized between 1927 and the 1970s, and these women were called effectives because of this way of thinking. Okay, so the American Association of Medical Colleges, AAMC, publishes every few years a sort of a guide, oversight of what we should be teaching medical students. And the one on genetics starts out with the eugenics movement, serves as the underpinning for contemporary misunderstanding and mistrust of genetics. And I hope you can understand from what I've been telling you why they might say that would be the case, because there are lots of concerns. So now getting back to where we began, genetic technology, if you think about it, bifurcates medicine into two branches. Okay? One is precision medicine, to make people better. And the other is eugenic medicine, which could become genetics to make better people. Okay? So, where we're going to go with this, I don't know. But I think that it's interesting that this movie, Gattaca, really which was not a very successful movie from back in the 1990s, really captured these two ideas in two scenes. One of the scenes occurs in when the parents are in the office of the genetic counselor. And it says, Maria, your eggs have been fertilized with Antonio's sperm, and we have performed an analysis of the resulting pre-embryos. After a screening, we're left with 2 healthy boys and 2 healthy girls. Which one would you like, basically? Okay? So that's one way of asking a question. And then later, the son who did not have a genetically determined birth, he's looking for a job. And he says, my father was right. It didn't matter how much I lied on my resume. My real CV was my cells. Why should anybody invest all that money to train me when there are 1,000 other applicants with a far clearer profile? So those are two totally different ways. One is using genetics to make informed decision-making, to give people information that they can make decisions with. But it's not eugenics in a negative sense, like this social choice, the social philosophy is. So, and just one last thing that I think epitomizes actually the way to think about positively what we should be doing. So here's a paper that just came out a couple weeks ago, or a couple months ago, that provoked the headline in Newsweek, a new genetic test could help determine children's success. Okay, that's genetic essentialism. That's not what the paper was about, really. But that's the way it's presented to the public. The title of the paper was Genetic Analysis of Social Class Mobility in 5 Longitudinal Studies. So it was not making any claims at all in the title. And the paper goes on to say, okay, genome-wide associated studies have discovered molecular genetic associations with education, a determinant of social class, and we found that people with more education-linked genetics were more successful in terms of education, occupation, and wealth. These are small differences, and the authors emphasize that there are a lot of potential questions that still need to be answered, but they end the following way, which I think is just how I would like to see us think about genetics. They say a long-term goal of our socio-genomic research is to use genetics to reveal novel environmental intervention approaches to mitigating socio-economic disadvantage. As we learn more about how genetics discovered in GWAS of education influence processes of human development that generate and maintain wealth and poverty, we can identify specific environments that change those processes. Ultimately, this research approach can suggest interventions that change children's environments to promote positive development across the life course. That's a positive way of thinking about the most positive, which I am. So I'm out of time, or well, I may not be out of time, but I'm out of things to say. So thank you all very much for your time.
Any questions?
Okay, I guess it's working. It's fairly obvious that the Tuskegee experiments and other experiments based on eugenics in the early 20th century turned out very badly. And I think that's universally acknowledged. At the time, though, perhaps you can correct me, it was thought to be very forward-thinking, scientific, modern, proper way to deal with, you know, the fate of the human race, and it didn't turn out quite the way they expected it to. How do we know that what genetic engineering is doing right now is not exactly the same thing?
So the question, everybody here, is the first part about the Tuskegee experiments in the early part of the 20th century. Some people will thought were positive and for betterment of humankind. Turned out not to be the case. And how do we know that now the stuff on genetic engineering isn't going to eventually be seen, let's say 25 or 30 years from now, the same way as being really bad stuff? So there are two things. The first thing is that the Tuskegee experiments were not about genetics. A lot of people don't understand that entirely. What the Tuskegee experiments were a group of public health people trying to see what happens if you have syphilis and you don't treat it. And so they were just following these men who were African-American sharecroppers who had syphilis, and they weren't treating them. And even when penicillin became available to treat syphilis, they told them that they were not candidates for that treatment. So this was deliberately,
They
you know,
gave them
huh?
They gave them syphilis.
Well, not in the Tuskegee experiments. In the Tuskegee experiments, there were other experiments going on in the 1940s in Guatemala, where people wanted to study a variety of diseases, sexually transmitted diseases, and people in Guatemala were actually given the diseases purposefully. But those are not genetic. Those are different than what we're talking about now. But the point that you're making is absolutely, I think, correct. That is to say that 30 years from now, we may look back and say, oh, this was bad stuff. We ought not to have been doing it. But one of the things that happens is that the more risky a research program becomes, like for instance changing the genetics of embryos, the more people should be involved in trying to decide whether or not we want to do it. So in what's happened in the United States, I don't know about other countries, but often the decision-making process for these riskier kinds of research programs involve not just the researchers and not just an institutional review board overseeing the research and not just the NIH funding the research, but also involve public groups as well to get feedback. And if we all agree, or most of us agree, we should go ahead and do this research, understanding what the potential risks are and the lack of certainty that we have about what the outcomes are, at least we can say that we did this in an open-eyed manner. Even if it turns out 30 years later we decide that it was wrong. So I think it's a really important question.
If one wanted to participate in this new program, or they're looking to get more applicants for the 59, for the program for the 59 something, how does one go about doing that?
Yeah, I don't know. But I'll bet you if you go to the National Institutes of Health website and you look up All of Us, which is the name of the research program, they will, if they're still recruiting, and I think they are, they would have a way of, a link you could put on and say, hey, I'd like to be, part of this.
Thank you.
Okay.
You know, I'm subject to, I haven't found a conspiracy theory I don't like. But basically, if you go to the UN Agenda 2030, or if you go to the Georgia Stones, there seems to be this kind of this idea that bubbles up about reducing the Earth's population drastically. And so, and some of you can trail some of these, I think even some of the families of the people that are associated with the families way back when. So my question is, but then you hear all about vaccines and all these problems with the vaccines, like vaccines that gave just cancer in the 1940s, and then if you watch the movie Vax, how the guy who actually designed the statistical analysis came out and said this stuff really is dangerous. So to what extent should I be concerned that there are still forces that are trying to correct the world's population in their view? Or is it just not? Good question.
Well, so vaccines are an interesting subject and reveal in some ways how problematic it is to answer clearly your question. So some of you know that there's a claim that some people have made that measles vaccine is associated with autism. And one of the consequences of that claim is there are a lot of people who decide not to get their kids vaccinated. And the incidence of measles has gone up. Now the guy who reported that relationship, a guy named Wakefield in the UK, was a very well-respected physician researcher. And so that's why some people believed it. 20 years after, shortly after his death, and his papers started coming out about him and research about him historically, it turned out he was being paid hundreds of thousands of dollars by people who were opposed to the measles vaccine. So, by law firms that were trying to create legal cases against the measles vaccine and for large settlements. So the reason I mention that is that there's an awful lot of trust in science about what scientists do, and we can be fooled. And it can turn out that there are that we hear data and relationships now that we learn 20 years later were not the way things really are. So I think that but then we have to act upon what our best information is. If we assume that there's some conspiracy, then we're not going to act. We're not going to take advantage of whatever we might be able to take advantage of to improve human health. It is clear that we live a lot longer now than we lived a few hundred years ago. And the world's population is getting very large to a significant extent because of the success of modern medicine. So does that mean the success of modern medicine is a problem because it's exacerbating, population growth?
Well, even I'm not, then I read stuff like yesterday I read something about the American Indians, the buffalo culture. They just lived on buffalo meat, period. And they said that, again, I don't know how accurate these people are, but they were saying that these people had more centenarians than any other population ever.
Yeah.
And so when you hear that we live longer, I even question that. You hear the Eskimos live incredibly healthy. The Maasai were incredibly healthy. And then you find out that the American people are some of the most unhealthy people in the world. And I mean, just as a thinking person, I say, I need to examine what I'm being told.
We are unhealthy by some criteria related to other people. But even that unhealthiness, we still live way longer than we lived 20 or 30 or 50 years ago. You can see it.
Hey, I got the mic. Andrew Wakefield is alive and well and lives in Austin, Texas. He's spoken Dallas.
I don't think so. Must be a different Andrew Wakefield.
What?
Must be a different
No,
Andrew
he's the one
Wakefield.
who is in the facts film and stuff. Another thing is the measles outbreak you spoke about. Those were the breakout. Those were amongst children who were vaccinated.
Well,
So, you know,
That's provable.
yes. My question was about epigenetics.
Uh-huh.
Can you give us a little information on that? And what is junk DNA?
okay, so let's start out with junk DNA. That was the last thing. So before the human genome was actually deciphered, it was clear that there was a guess that humans would have a tremendous number of genes, maybe 100,000, maybe 150,000. And then we actually got the human genome, and we discovered that only about 5% of the genome becomes things like proteins. And the other 95% people thought didn't do anything initially, or didn't know what it did. I shouldn't say they didn't think. Some people thought that it was 95%. That must have been really important. Some people thought that, well, that's just junk DNA. And so that term got picked up by the press, and it was around there for a while. And so junk DNA was all the DNA that was not coding for proteins or other specific molecules that were functional. What's happened is it turns out that all of the, not all, but most of the regulatory elements that control the genes are in that area that's called junk DNA. It's in those other sequences that are nearby genes but not part of genes that control when they are expressed and how they are expressed and whether they're overexpressed. And a lot of the recent research association with the potential for developing cancer in certain instances is the idea that some segments from those areas that are not expressed all of a sudden move and disrupt a gene that is expressed, and that disruption causes aberration and can lead to malignancy. So the so-called junk DNA has become central to understanding regulation and evolution and physiology, not because it leads to specific proteins, but because it controls what's going to be expressed and what's not going to be expressed. And what was right before junk DNA? Epigenetics. Okay, so this is what the reason why you have these epidemiological laws. So think about epigenetics. So let's think about cancer. In order to get cancer, you have to have a bunch of different genetic changes that occur in your genes. Or those genes, those are not inherited changes that occur. Those are changes that occur because of your lifestyle. So you may have, let's say that it takes 5 genes, 5 changes to make skin cancer. And you're born with one of them already changed, inherited. Then you have 4 more, and those changes can occur because you spend too much time in the sun. So now you've changed the genes, you've influenced the genes through sunlight, okay? And now you've created enough situations where you have all 5 of the cancer-related mutations, and now you get cancer. It's genetic. Only part of it is inherited. So those are all changes in genetics. Genes have all of these little markers on them, little modifications by things like different chemical groups. And those modifications determine the extent to which specific genes are on or off. those modifications are called epigenetics. That is to say, they're not part of the DNA sequence. They are modifications on top of the DNA that's there. So you can have a change, so you can have let's say a methyl, it's called a methyl group, put on top of a base, one of the DNA bases. And the presence of that group can now change what that gene is going to do. So you can have changes in the behavior of genes either by mutation, where the actual bases are changed, or you can have those changes by different chemical modifications. And the epigenetic one, and the epigenetic modifications are just as important because they regulate what's going to happen to a large extent. Is that? We can argue about the Wakefield.
I just was curious, did you see the piece last week on 60 Minutes about did anyone, about the polo player in Brazil who has been the world's best polo player for 20 years?
Yeah.
Did you see it?
No.
And his horse that he used for forever broke its leg and it devastated him. So before they put her down, they got her cells, etc., and cloned 13 horses just like her.
Okay.
And they all get along. They're all, it's all harmonious, and they're both all excellent. They're all excellent in this competition. And I thought it was just simply fascinating to see this recreation in the laboratory. I'm not real sophisticated in this area, but when we were talking, I could be brought back last week. It was most fascinating.
So there are, the comment is about the recent cloning.
And the cloning person who's doing this lives in Texas.
So the very first cat that was cloned at Texas A&M, and they called the cat Copycat. And this was a while ago. The only thing was that Copycat looked much different than Mom. Because it was had that fur pattern, which is multicolored. I forget what it's called.
Calico.
Calico, and so it can it's influenced by epigenetic modifications. It's not just dependent. So, but now they're all over the world. I don't know how much in the United States, but in many parts of the world, cows, horses, pigs, all sorts of animals are being cloned. The only thing that's not being cloned yet is us, and one of the reasons probably is that the technology involved in cloning often fails. So if we tried to clone us, the question is, what would we do with all the mistakes? And so we haven't cloned us. But the technique used for making the 3-parent embryos, that's the same technique as for cloning.
I heard once that these animals that were cloned, the little thing that determined their age, the telomere,
The telomeres?
it started short, and it was transferred to the clone. And
So there are questions about whether or not you're cloned, whether you start your age at the age of where you were cloned from, or you're reprogrammed. Yeah.
Epigenetic modification to cure disease and prevent disease. On a clinical level, how does it work? For example, if I had a health problem or knew that I inherited a health problem and I wanted to be treated on that level, how would it work in a clinical setting? Do they take samples of tissue and change the gene? How does it work?
It doesn't work. We can't do that yet. The closest we can get to treating you by genetics is to take some of your, let's say that you have a disease called, you remember the bubble babies? Okay, so those kids have a defect in an enzyme called adenosine deaminase. And that enzyme is produced, I think, by one of the blood-borne cells. And so they can get the stem cells from a kid, take them out of the kid, and in vitro, they can use a virus called adenosine, called the adenovirus, to transfer a correct gene into those cells. So they haven't changed the lack of the correct gene that was there to begin with. But now they've put in a gene that can make that enzyme. And then they take those cells that have been modified and put it back in the kid. And now that starts making that gene. So the goal of CRISPR would be to take those cells out and fix the gene and put those back in. We don't know how to do that yet. So all of these technologies are all, that's why the idea of racial hygiene to some people is so appealing, because there you're not fixing it, you're just getting rid of it. Okay, and so we don't have the technology yet to fix things very well.
One last thing. Do you think that these point by points with the racial hygiene through the years, now we saw it, but the threat of it, do you think that may be a part of just hindering research in health-related issues, or is that just a fine line that very spike degrees they're having to watch?
So the question is, does the concerns about racial hygiene impede the research? I think that the biggest impediment to research in the United States, anyway, is the question about the embryo, because all of this research surrounds the embryo, and we have a community that's as divided about the embryo as we have divided about our politics. And so the net result of that is that there's no embryo research, and until there's some, and I don't see that changing in the near future. So.
You say sunlight affects the DNA, and I read that being happy affects your DNA. Does it make it, is the DNA just waiting there to be activated, or is it, how does that work? And also, does sugar activate, do anything to the DNA, and alcohol?
Okay, so those are all good questions. What does stuff to the DNA? First place, being happy, I like that one especially, because being happy makes your immune system work better, okay? And being stressed makes your immune system work worse. So when you're stressed and your immune system doesn't work, you're really upset about something, very often you get a cold, right? And the reason you just got that cold is because the immune system, you always had those viruses that caused the cold, but your immune system was keeping them under control. So I don't know that being happy has a direct effect on the DNA, but it has a direct effect on the physiology and everything that it interacts with. The sunlight has radiation in it, and that radiation can directly damage the components, the chemical components of your DNA, and cause them to breaks or mutations that then cause a deactivation of those, or overactivation of those cells. So one has a very indirect way of impact, but the other is very direct. Sure.
3 short questions. With Angelina Jolie, that's the reason she had the mastectomy, because they found out she had genetic
Yes.
that her mother had.
Yes.
So that's the preventative
Yes.
way that genetics is being used.
Right. So Angelina Jolie and other people, other women who have discovered that if they have the BRCA1 or 2 mutations, and especially they're sensitized to looking for that now if they had an early death. If your mom died at age 40, let's say, from breast cancer or uterine cancer, it's and then about 99% you can get rid of the potential for developing those cancers if you get rid of the cells, the organs that are affected. So, yeah.
Okay, I think I read that the Catholic Church now approves stem cell research. Is that correct?
So the question is about the Catholic Church and stem cells. The Catholic Church has never been against stem cells because we have two different kinds of that are from the adult. We have two different kinds of stem cells in us. One kind of stem cell is the cell that is constantly within us repopulating our tissues. So that, you know, your stomach, for instance, is turning over because it gets damaged. Well, there have to be cells that replace those, and those cells that are your skin, you know, is constantly turning over. So there are stem cells that do that. Catholic Church is great for stem cells. The Catholic Church is very strongly supportive of research. They just don't want stem cells from embryos. Okay? That's where they don't want the stem cells to come from.
Okay, and you said that in the United States, we do not have that research for embryos at all.
We don't have it supported by federal funds. So what happens is that in the United States, we are a very heterogeneous group. We do not have top-down regulation. Even if you think about something that's so controversial, like abortion, there are some states that try to stop it, and there are some states that totally embrace it. So it's a very state-by-state kind of thing. So California, as soon as the federal government said no embryo stem cells from embryos, California started a robust the California stem cell research program, and all the researchers who wanted to go, you know, they all went to California. And so we have a great cancer program here in Texas.
Will other countries do this research?
We are the only ones who have as the kinds of bans that exist. And we have a very thoughtful oversight mechanism that we use to talk about things. There are a lot of countries where the research is going on and we don't really know what oversight it is. Britain has the best system actually. So Britain knows every embryo that's ever been artificially made. Who made it, when it was made, who the parents were, etc., etc. In the United States, we probably have 500,000 embryos in freezers with lost connection to who the parents were who made those because of the way our system is.
I heard of an experiment just recently on mice where they took a set of mice, they introduced a smell of some kind, and then immediately after they introduced the smell, they shocked the mice. And they got to the point where they could just introduce the smell and the mice would react as if they were about to get shocked. And then those mice had babies, and they introduced the smell to the babies, and the babies also reacted as if they were about to be shocked. But the babies had never gone through the experiment. So there was some kind of inheritable DNA that was not directly manipulated in the DNA. So how does that feed into your genetic experiment?
Beats me. I know. That's a kind of experiment where there's certain results in experiments where I sort of bracket the experiment. It's not that I say that I disbelieve it. It's just that I say I want to know more. I want to see other people do it. Because if it doesn't make sense to my general way of thinking, then I'm not going to just change my general way of thinking because somebody says some kind of a research result that it differs from it. But that's an interesting finding. Scary, actually.
Yeah.
Yeah, is it not true that genes interact with other genes in mysterious ways? So if you fool with one gene, you may be doing unknown damage, in fact.
Yeah, so this is a very important issue, that genes do interact with each other. And so for instance, there are many different strains of mice. And you can make a genetic change in one strain of mice that will have profound effects on that mouse. And you make that same change in another strain, it'll have no effect. And so there are a lot of interactions between genes. They turn each other on and off. And one of the problems with the whole idea of engineering embryos is the unanticipated consequences. It's always the unanticipated consequences that do us in. So.
Also, apparently the Chinese are, from what you said, are developing gene therapy and so on, whereas the US is not. Is this the case of Chinese capitalism trumping American religiosity?
Well, I think the question is what's going on. I think that people in the United States are trying. That three baby kid, the first one, was an American physician. He recruited Jordanian parents and went down to Mexico to do the experiment. So the thing is that we do. I'm sure that the stuff that's going on in the United States, it's just that it's harder, and it's because there's less support for it. And the Chinese, we don't know what the herbicide mechanisms are. Yeah, so yeah.
So where do you see all this gene research and gene therapy leading? I mean, what's your expectation factor?
So my expectation is that before long we'll be able to make genetic changes to people's cells that we take out of them and put back in those cells back in them and have impact on them. My expectation is that we're not going to be engineering embryos for any time in the future. It'd be a lot easier to just do pre-implantation genetic diagnosis on embryos that are formed naturally, and then make that whole process more efficient. And then sort of like the in that office, you have 2 boys, 2 girls. Which one do you want?
My question about something you said earlier, perhaps misunderstood what you said about the impact of genetics, inherited genetics. My wife's mother, father's family.
Speak up.
My wife's father's family, my mother's family, had a terrific problem with cancer. And my wife's mother had 6 children. And out of 6 children, 3 died of cancer, which one of them was my wife. And one other had breast cancer, because she's the oldest daughter. And it went into remission. So my take on that is genetics did have an impact. And you said something about something else maybe.
So what happens is that, let's think about, I said that you need multiple changes, genetic changes, to get breast cancer. And one of the key changes you have to get is changes in this gene called BRCA1 or BRCA2. Now, if you're born with that change because your mom had that, and it was part of your genetic transmission from your mom, you're much more susceptible to getting breast cancer than if a mutation from what you eat or your interaction with your environment has to cause that. So we interact with our environment, the radiation and the food we take in and everything else has potential impacts on our genes. And those happen over a period of time. But if we're born with it, and so that's what's happening. And that would be the typical explanation.
That's what I thought.
Right. I think you're exactly right.
I never seen a family with that high ratio of
Right.
children with cancer.
That's exactly right.
What is the possibility or the likelihood of combining the superior traits of each major gene pool into a master superior race, genetically speaking, or breeding a super race, reminiscent of Grinnell? What's the odds on that?
So that's always the question that bothers the people. Could we have a super race or something? So here it's a good example. You and your twin, if you have a twin, are very similar in height. About 80% of your height is controlled. genetically by about 1,000 genes. Your intelligence is controlled genetically by about 1,000 genes. So trying to engineer specific things like height or intelligence or a lot of other, related things that have so many genetic inputs is a very unlikely outcome. And they will interact with each other. So I'm not worried about any genetic super races coming along anytime in the near future, personally.
And you're saying the vast majority of changes in the genes occurs after a person leaves home, like 85% or something?
No, what I'm saying is that the vast majority of the mutations that occur are not what you got in your genes to begin with, although there's some that count a lot, but that you get later on in your life that occur
After
just
you leave home from the effects of the environment, like too much sun or
right, exactly.
too much alcohol or too much cocaine. Or,
Yes, you got them all.
the whole lot.
I suspect in some ways we're looking at this completely wrong. We're looking at it, look at this guy, his DNA. We want the best possible DNA we can get. He's got bad genes, it's going to happen to him. But we evolved in groups of 25 or 30 people. And so really I think we evolved as gene pools. And we had a couple of big guys, a couple of little guys, a couple of, all different types of personalities. And that group of people worked together. And I suspect that, let's say, the sickle cell gene, it's a bad gene. It's a bad gene perhaps for the individual, but for the group, that sickle cell anemia gene has benefit in case there's a malaria epidemic. So I think looking at it just as an individual, if we start eliminating bad genes, we don't know that gene somehow protects us from something. So should we think about this as a group rather than just as an individual?
So one of the things you're saying is relates to one of what's become major research programs, and that is that you identify a gene that for a large number of people has negative consequences, and then you discover that gene in somebody where there are no negative consequences. Now all of a sudden you want to know, well, how come? I mean, this gene is supposed to have negative consequences if you have this, and now you're not seeing them. So that sort of reverse way of looking at what genes, if you've inherited, we're all products of natural selection. If we have genes that are detrimental to us, one presumes that they must at some point have had some benefit. And so the question is, well, what was that benefit, or what is that benefit? So it's sort of...
Yeah, I'm sort of saying maybe that bad gene protected us from the plague.
Maybe.
The plague only comes along every 500 years.
Well, yeah, it's maybe. Well, all of us have to struggle, with our weight. And the reason why is that our bodies evolved to have relatively little nutrition on a regular basis. We used to eat a lot, and then we would have a famine. Then we would not eat, maybe for a week or 2 weeks or whatever, or very small amounts of food. And we come, and now we live in a modern society where we have so much to eat. Well, that's a constant struggle. Yeah.
I apologize, I've not read that much about genes, but is there a genetic system that enables one to have musical ability?
Yep. The music gene. I'll bet you that there are genetic influences on musical ability, and I don't know how many genes are involved, but I bet you there are. I bet you, So, yeah, why not?
It's probably a full scale.
Yeah. Good. Did you all get that back there? Was it C clef or F clef? Never mind.
A little off topic, What kind of courses do you teach to your students, and what kind of courses cover the topics of genetics?
Well, I teach an introduction to biomedical ethics.
Biomedical ethics.
So genetics is one of the topics. But end of life, beginning of life, genetics, enhancement, justice, those are all major issues in biomedical ethics. So I teach that, and I also teach about what's called research integrity. So it's sort of related to kind of professional ethics, but when you do science, there's a lot of ambiguity, and most people don't realize how ambiguous scientific decision making is. And so it's important for students to understand that and realize, what they're dealing with.
Any other questions?
All right, I'm out of answers if you're out of questions.
You get to sit down. Now it's your turn to talk, so you get 5 minutes, see what you want, and respond to the speaker had to say for or against.
Well, one thing that you stated as a fact I would like to refute, and that is that all brain-dead people are on life support systems in hospital. I have met some walking around amongst us. And as far as genetic material collection is concerned, the only one who's ever collected genetic material from me didn't give a damn, he swept it away, and that's my barber. And my parents raised us to know that we had the best of genes, and I've always made sure that the only thing I ever bought was Levi's. Now, I think, you did your percentages. I think a much greater percentage of all of it is in consciousness, which includes all of our emotions, all of our beliefs, and everything else. And one little example of that, I was reading years ago that in Harvard, some guys did a little experiment. They took a guy and tied him to a chair and told him they were going to cut his veins and make him bleed to death. And they rubbed an instrument on him and made him think that. And then they kept dropping lukewarm water on him. He wasn't bleeding, but he died because of his belief, his fear. There's that Bible statement, and that which ye most greatly fear shall come upon thee. I think that whole percentage, I think consciousness is a much greater percentage of it all. They're just an example of things that are so strange. I remember reading about this man who went through school as a normal kid and had average grades and played on ball teams and everything. Got out of school and got a job and married and had kids and had a 2-car garage and the whole bit. And then he needed to get a brain scan, and for some reason, they found out that he had in his skull only a brain stem. He did not have the bulk of his brain. And there have been several people found like that. And scientists have absolutely zero explanation for that because it defies all their scientific beliefs. I have a real problem with abortion because that child is a living child inside the mother. I was down at the in Tampa at the celebration similar to Mardi Gras, the Gasparilla Parade, and I was standing beside this couple with their little boy, and the little boy and the father seemed to have this very, very close relationship. And I remarked that to the mother, and she said, yes, we had a home birth, and he was in a breech position, and my husband had to reach inside of me to correct him. And when he did, our son grabbed hold of his finger and held on tight for a little while, then reached up and pulled his own shoulder down and came through, and they've been very close ever since. I knew another couple, when she was very large and pregnant, she called her husband over and said, put your hand here on my tummy, and he did, and the child pushed against his hand. She said, now move it somewhere else, and he put it over here, and the child pushed against over there. The child was communicating in the womb with the father. As far as sunlight is concerned, you don't want to eliminate all of sunlight because it is absolutely necessary. That's where we get natural D3. There was one doctor I read about who got she worked in an emergency room for years, and she got a breast cancer, and she knew the medical profession and chemotherapy and all of that, and she just absolutely would have nothing to do with the medical profession. And she did all natural means, and one thing that she required was that even when she could barely walk, her husband took her outside into the sunlight for at least a half hour a day, because she felt that was essential to her health. And she recovered completely. There was a video that was on one night, and a friend of mine was recording it about 2 a.m., but it was taken off the air before it was finished. And it was an interview by a man who had worked his life in the pharmaceutical industry. And he started talking about drugs, and he said, all the pharmaceutical drugs, none of them are designed to cure a disease, because that would eliminate a source of revenue, a huge source of revenue. He said they're all designed only to manage your disease, which is the terminology they use. And he said also, every drug has in it other chemicals, so that if you take that drug over a long period of time, you're going to develop another disease for which they have another drug, and then another and another and another, and that's why older people are on 6, 10 drugs. They're being poisoned by that industry that makes billions out of us as the suckers that they make all the drugs for. There are a lot of natural remedies, and I would pursue those first. That's just my viewpoint on it. Mel Gibson took his father down to Panama, where they're doing extensive development in stem cell therapy. His father was way into Alzheimer's, and he took the treatment, and he came back, and he says now his father's cracking jokes and just as bright as he ever was. So stem cell therapy could be a very good thing. Again, I think that consciousness, like happiness, like joy, like doing kind things for other people and for animals, all of these giving things, all of these build up our immune system. Plenty of rest, don't get stressed. All of these things that we can do will build up the immune system probably a lot more than drugs can. And we should always work on those positive aspects of life. And just remember that statement, and that which ye most greatly fear shall come upon thee. So watch your fears and develop a positive attitude and know where your strength truly comes from.
It seems that science has an attitude that if it can be done, it should be done. Now, I understand that there are biomedical ethics, and I'm sure there's scientific ethics in a lot of other areas, but there seems to be this prevailing idea that if we can do it, we ought to do it, and we ought to see, you know, what the results are and stuff like that. But what's been presented so far tonight is a fairly limited set of what can be done with the purported aim of therapeutics, of seeing what we can do to help people to cure disease and such like that with genetics. That may seem kind of reasonable. On the other hand, Joel Wallach, who is a well-respected veterinarian, doctor, whatever, he has done something like 30,000 autopsies on animals. And he said, I forget exactly what the disease was, but it was something like cystic fibrosis or some horrible, horrible disease or condition, that his approach to curing disease and things is nutrition. Like, he will figure out what nutrients are necessary to deal with this problem, and your body knows how to use those nutrients and cure it. He said that he could turn this disease on or off in certain animals by simply withholding or giving them a certain nutrient that was necessary to their development. So then you have to ask the question, Is it necessary to deal with genetics when perhaps all that's necessary is a person getting the right nutrient? That's one approach, or another approach to dealing with a lot of these things. And the nutrient approach is very successful. The other thing to think about is that, you may be thinking about therapeutic approaches to using genetics and how can we help people. But then what other people might be interested in using genetics? Like if you took the military and said, we're going to give you an unlimited budget, which they basically got if they really want to do something, wouldn't they think about creating super soldiers? Not just, let's keep everybody healthy, but let's get soldiers that can run at 30 miles an hour or 50 miles an hour. Soldiers that can jump enormous distances, climb mountains, with incredible speed and ease, have an incredibly keen eye where they could, hit something with a rifle at ridiculous distances. And then you have things like, why not create a human being that can swim in water and doesn't need oxygen? Who's got gills? Somebody who can swim around underwater and, just like fish. Or why not find the genes for like an eagle or a condor and grow somebody with wings so they can fly? And when you start thinking about things like that, you can start looking at ancient Greece where they supposedly had minotaurs, a man with a bull's head, centaurs, a horse with a man's body, and who knows what else. I mean, if we're dealing with genetics, which determine how a thing grows or what it grows into, what would prevent anybody from developing those things if they've got the money? I mean, you get somebody like George Soros who's got all kinds of money. Maybe he's working on developing some kind of super soldier or super smart person that can help him achieve his agenda. So when you think about those things, the idea of if we can do it, we ought to do it, raises some very serious ethical questions. And I suspect that no matter how many good intentions are involved in all this genetic engineering, we're going to look back in 50 years and we're going to realize that there were some things that we missed.
Yeah.
I agree with Ken in that our speaker's presentation gave some focus on how genetics would go. He did answer the question, however, about these bizarre people that you were talking about when he said how difficult it is just to control the height of a person. But my comment is just a layperson's comment on the complexity of the issue. A few months ago, I was in an argument with a friend, and I ended up Googling, when does life begin? And interestingly enough, a fundamentalist Christian biblical scholar had an opinion that I did not expect. He said that it used to be easy to determine because in Genesis it talked about God breathing life into the person. And so we believed that when they were born and took their first breath, they were a person, they were alive. But it's gotten complex because now we know with scientific discovery that children born early, all of, what am I trying to say? Premature children, they can do so much to save children that are born early. And the question became, when are they viable? And so that's basically, I'm having difficulty remembering all I was going to say, but it's a complex issue, and genetics is here to stay. It's not going to go away. You know, it's how do we use it, and I guess that's basically my statement.
I'd just like to review I think a very important point. There can be genes in the human genome that seem to be bad or are bad when they combine in a certain way, and if we eliminate those, there may be diseases that have come and gone, you know, over the centuries that these genes give us some protection from. So everybody's, you know, the people with these bad genes survive and humanity goes on. I think that's the thing with sickle cell anemia. If you have the recessive gene for sickle cell anemia and the good gene and a regular gene, you get immunity or some immunity from malaria. So that's why the sickle cell anemia gene has never gone away, because there was benefit to it. So we just have to be careful what we get rid of, because we really may not know what these genes that are in the population may have some real benefit in some other situation, exposure to some particular germs. The other thing I say is this: my perception about medicine is that I've become aware. What I thought is, I used to get headaches, and somebody told me to take iodine. I took iodine, my headaches went away. They said everybody is low on iodine, period. The Japanese get 100 times more than we do. They don't have all sorts of different problems that our other people get, because we're fundamentally low on iodine. So anyway, I take it and fix it. And then I had an arthritic problem in my elbow, and I read some old slave medicine, and this came from a doctor, and I took turpentine, believe it or not. You know, it says fatal if swallowed. I took some of that, and it went away. But I got it from a doctor, you know, who's, you know, outside the country of this old slave remedy. And so what I've become to realize is that doctors really aren't doctors, as I understood them to be. Doctors are really administrators of accepted medical practice. Yeah. So if you go to a doctor and you have a specific disease and he says, you know, yeah, I want you to take some turpentine, or I want you to take, you know, he can't do that, because even if it worked, he can be sued because he was outside the prescribed remedies for that disease. So really, doctors administrate medicine as it's understood by, I guess, the American Medical Association. So the last thing I would point out is, how did we get there? And like I said, I had never met a conspiracy theory I didn't like. Well, here's another one. Basically, you used to have all this old kind of medicine, and however, the little bits I use, you know. But what happened is that the Rockefeller Foundation came in with donations to all the medical schools. And so then they put their people on the boards, and then they said, well, we want to practice allopathic medicine. And so basically all that stuff was pushed aside, and we have allopathic medicine now, which is not turpentine or iodine, but it's just you have a symptom and you have a corresponding
Pharmaceutical.
Expensive pharmaceutical. So I'm very suspicious of medical, and by connection, I would probably be very suspicious of any results coming out of. I would like to know who's financing, who's sponsoring all the genetic research. I want to know who's paying for that, and that would be. I'm just very skeptical. So thank you very much.
I'd like to thank you for that speech. It's very interesting. You know, I think it really seems to boil down to, is it nature or nurture? And I tend to agree with what you said, that the genes are a part of it, but then what happens, what we do later in life, really counts for a lot. And I think you see examples of this. For example, some people have natural athletic ability, but it's training that turns them into super athletes. And there's a case in London, for example, the London cabbies have to take what's called the knowledge. Well, what that is, they drive around on a little motor scooter, and they have to learn basically every street in London. So they can, you look at how many streets there are, and it takes them almost 2 years to get this so-called knowledge. But what they've seen is it actually increases that part of the brain which has to do with memory and location, actually. So they proved that. So I think that, yes, a lot really does have to do with, as you say, how you apply your stuff. And I think the other evidence is where you see rich kids, I've seen, who turn out to be quite dumb, because why would you study hard and improve your brain power? Because why would you? I mean, why would you beat your brains out, as I did, learning, Lagrange equations and goodness knows what, also, differential analysis. I mean, really, it's difficult stuff if you don't have to. And so I think, yes, what we apply ourselves to really does count for a lot. And so I think sometimes people are born with different traits, and it's a question of applying yourself. Now, with respect to gene therapy, yes, because as you say, there are definite, as you say, you're born with certain defects, which, I mean, if you can get rid of them by CRISPR and other techniques, are really valuable. But certainly, if you can eliminate certain cancers and certain other diseases, maybe even Alzheimer's, that probably has. But it's this problem really is, as you said, it's this combination, the effect of combination of genes which makes it so difficult. So, these are very difficult problems, but to deny science really, to me, is, I'm a scientist, and, really the way to go is, of course there is junk science, and there's people who are charlatans in science. I mean, when I did my research, I realized afterwards I could affect the whole thing, really, because nobody knew what, my professor certainly didn't know what I was up to. It was only when he read my thesis he realized what I'd done. He had no clue, so I could affect the whole thing. So, there you are. So let's hope progress will be made, in these advances, because really, I think that's the way we, as a society, progress. If we actually do and get empirical knowledge, do find out what works. And of course, there are these, things that you try and seem to work. Well, it may be connected, and maybe not. It may be just your mind doing the mind over matter, really. I think a lot of life is mind over matter.
First, our speakers, thank you so much for coming out. This has been a wonderful talk. Thank you so much for bringing this material to us. It's just been really refreshing, and it's been very informative. I really appreciate you coming out, and I'm going to try to condense this in 5 minutes and keep it organized without turning into a deer in the headlights. I tend to do that. As far as religious dogmatic thinking impeding upon medicine and the advancement of knowledge itself, I don't believe things have changed that much since the 12th century. I myself am pro-choice, and you good, Margaret? I myself am pro-choice, and I do not understand how this dogmatic thinking could affect the progress of medicine that could help save lives within itself. And I can't claim or even speculate as to what's going on, why those things are at odds with each other, but oftentimes they are. And I do agree with John that some aspects of this do come back to nature versus nurture, whether this is inborn traits affecting our genes or things in the external environment. And I agree that that does come back to a nature versus nurture debate. And just on a personal note, I have seen things that there is no test tube in the world that would fix that. And I know a lot of people like to speculate and imagine like life is a Saturday morning cartoon. We cannot engineer people. And I'm really pleased to hear somebody come out and say, hey, we have 1,000 variables to any given trait. And I don't think that medicine within itself is that insidiously organized. In fact, I believe it's quite the opposite. But on the topic overall, I am so glad to hear that there are biomedical ethics classes taught. I had no idea. And I wouldn't have known if you hadn't come. Thank you very much.
Anyone else? One of the last ones standing? Well, I want to thank our speaker for doing a great job here tonight. I don't have much to add, but I learned a lot from what you had to say, and I have to say that it's good that someone is looking at this type of endeavorment because it helps society. It makes us better and it produces a better result in the future. So I applaud that. I also like the fact that you're in ethics research as far as how that is done, and eventually maybe we get the thing and do it right. We don't want to create a super race, that's for damn sure, but we want to make things a little better for everybody that's here. So anyway, our speaker gets the last word. Come on up here. You get the last word.
Well, thank you for all the comments. I guess I just a couple of thoughts. The first thing is that I'm not a physician, but when people ask me about medical interventions, I tell them that my first advice is to meditate every day, and my second advice is to do yoga. So I'm a big believer in natural, sort of naturopathic relationships of our bodies with the world. That's the first thing. The second thing is that I don't think that that it's technology, not genetics, that the military and others are going to use to change the way we are. It's very hard to change the genes which come from millions of years of evolution. So engineers don't really want to intervene in systems that they can't understand. But when it comes to building a system from the bottom up, like creating, there are these some of the innovations that we see on TV and in the movies of people who have these adaptations that are technologies that make them run faster and fly higher and shoot faster. I think that's all going to come personally from technology, not ever from genetics. That's my own prejudice. And then the question of when life begins is a really interesting one to me, because is there some relationship between life and death? If you say when does life begin, does that also entail asking when does death begin? And it really interests me that the single cell that forms after fertilization, the life of that cell, the death of that cell, is just cell viability, one cell. As soon as there are a couple of hundred cells, then one cell dying is no longer the end of life. Now it's this developmental arrest. And that goes on for a human fetus until about the 5th month, when the fetus becomes complicated enough so that we have a vascular system. And now we can have cardiovascular death, but we still can't have neurological death for another few months after that, when we have a neurological system that So this idea of life beginning, this is an example of this idea about that I just explained to depth. What's happening is that it's changing over time, and you have to say what you mean by life if you're going to ask the question when life begins. And we use language all the time as if we knew what we meant. But we really don't exactly know what we mean, and then we get in arguments because we each have different assumptions about what it is that we mean. And then one last thing, and that is that somebody mentioned, and I really appreciated, that Andrew Wakefield is still alive and living in Austin, Texas. I did not know that. Andrew Wakefield, he left Britain. I thought he disappeared from Britain because he died. But he didn't. He moved to Texas, and he's living in Austin even as we speak. So whoever told me that, I really appreciate that, because now I know that for the future. So thank you all very much.
There you go.
Well, that's it for tonight. They keep us out here at 10:30. For the speaker, the forum meeting's over, so long next week.
Oh, really?